Firstly, before we dive in deeper into our research study, let us take a moment and understand the current scenario of worldwide energy demand. The worldwide demand for biofuel is continuously increasing over time due to the expected decline of fossil fuel, gasoline. The United Nations Sustainable Development Goals predict that fossil fuel will be replaced by biofuel in 2100. Thus, the industry has to triple the current output of biofuel by 2030 in order to meet the International Energy Agency's targets. To achieve the target, it is necessary to find a green and renewable alternative source for biofuel production. To date, a total of 153 billion liter biofuel comprising bioethanol, biodiesel, and biobutanol has been produced. Between the three types of biofuels, biobutanol is considered an advanced biofuel due to its lower autoignition temperature, less corrosive, lower evaporation rate, high energy release per mass, and blend with base fuel without separation. Okay, now let's talk about the general process of how biobutanol is produced. Here we can see a simple flowchart whereby firstly, biobutanol is mainly derived from the fermentation of sugars in organic feedstocks. Butanol is a byproduct of this process in addition to ethanol and acetone. The process, known as acetone butanol ethanol (AB) fermentation, which uses the microbial species Clostridium acetobutylicum. Biobutanol, on the other hand, has the potential to be applied as future transportation fuels, but then it is still not economically feasible as bioethanol due to several constraints. Therefore, this brings to our problem statement, whereby the main problem rise from biobutanol production is its toxicity. For instance, at high concentration of butanol, it causes inhibition towards the Clostridium species, which leads to denaturation. Not only that, high cost is needed for product recovery. To further support the necessity of conducting this research, these are some of the examples from previous studies that portrays the constraint caused by butanol toxicity, in which the maximum inhibitory concentration of most of the Clostridium species are below 15 Gra per liter. In the same context, our solutions are more derived towards engineering-based. We as engineers, we want something that is cost-effective, easy to handle, and provides better productivity. All of this can be achieved through adaptive laboratory evolution, ALE method, whereby it's actually a technique of improving the strain through serial cultivation with different pressure until it can be adapted to end goals we want. In order to achieve our goal, ALE method was applied to our locally isolated Clostridium acetobutylicum YM1 strain using serial cultivation to enhance its tolerance under specific selective pressures. For instance, in our study, we emphasized on improving the strain tolerance level by subjecting to high biobutanol concentration that starts from 15 gram per liter and end at 23 gram per liter. Next, we will briefly discuss on our methodology. We start the adaptation with 15 GV/L because we found that this is the inhibitory butanol concentration for our strain. Once the strain has acquired the strength to sustain at that particular concentration, then we move up to next concentration. This is continued until the strain reached its limit. Then, once the best adapted strain was produced, it was scaled up in a 2L bioreactor using optimized TYA medium to assess its feasibility for industrial application. So now if we look into our result, we can see the success in adopting the ALE approach through the improvement of butanol production yield and productivity in adapted strain. But we can also see that the improvement is not very apparent. Therefore, we came up with a novel method that can enhance serial cultivation method during ALE, which is called activation approach. So how does this approach helpful? This method prevents overexhaustion of the strain and also to promote longevity of the strain to tolerate higher butanol concentration. As a result, we can see that the adapted strain it can sustain up to 22 Gra of biobutanol and produce 1167 Gra of biobutanol, in contrast with wild strain with negligible amount. This adapted strain of 22 Gra was further selected for closer comparison with wild strain. Here we can see that even when butanol was added to the medium, the adapted strain can produce high butanol without impacting the viable cell. In this case, butanol was not added to the medium, and the butanol produced was significant for a lab-scale result, which was up to 21.26 gram per liter of butanol produced. Besides, in terms of close comparison between butanol added and not added, it can be seen that the strain without butanol addition shows superior butanol production, productivity, and yield compared to the strain with butanol addition. On the other hand, scaling up strategy revealed high amount of butanol produced that further highlights its feasibility for industrial application for biobutanol production. As for invention advantages, our study has managed to enhance butanol production with significant improvement from lab scale to scaling up, and also butanol tolerance, in which there is 7 increment, which is by far the highest compared to previous study that only has 1 to 2 increment improvement. In terms of cost estimation, it is also proven how cost-effective employing ALE strategy in contrast to genetic-based strategy. In terms of commercialization potential, we have established a collaboration with a local industry, Free the Seed Company, which has expressed interest in utilizing our adapted strain for biobutanol production using rice straw waste. Additionally, we have an international collaboration with the Papua New Guinea University of Technology, where their copra waste is being explored as a substrate for the co-production of biobutanol and biohydrogen. In terms of novelty, our adapted strain, the first Clostridium species in the world that could tolerate butanol concentration up to 22 grams per ml. We also have our own method produced, patented, and copyright under MyIPO. Furthermore, our research achievements include winning a silver medal at the Malaysia Technology Expo 2025. Additionally, we were awarded a silver medal at AgroRoic 2023 and a gold medal at the Chemical Engineering Colloquium, recognizing the impact and innovation of our work. We have also secured a patent and copyright for our research study and established collaborations with industry partners, further strengthening the commercialization potential of our work. Here are some of our published papers that highlight on the scope of biomass and renewable energy, specifically in biofuel production. These are few major papers that we have referred in completing this research and slide presentation. That comes to the end of our presentation. Thank you so much for your attention.